So we have discussed about apoptosis, and as you remember, when we were discussing about apoptosis in the beginning, I told you that apoptosis are programmed cell death, right? But apart from apoptosis, there are other conditions also which are considered as programmed cell death. So now we are going to discuss those other programmed cell death. Programmed cell death, right? So programmed cell death, here I am going to discuss, there are two important topics which are to be discussed in this discussion. It can be necroptosis, number one, and it can be pyroptosis. So these are two important conditions where you will observe programmed cell death, right? So these are two programmed cell death which I wanted to focus upon, because examiner had asked this question earlier in exam कि programmed cell death without caspase activation is known as without caspase. What is without caspase? Necroptosis or pyroptosis? So answer is necroptosis. So whenever examiner says programmed cell death without caspase activation, see, without caspase activation, that will be called as necroptosis. So that was the question in one state exam. Without caspase activation, programmed cell death without caspase activation is called as necroptosis, right? And pyroptosis with caspase activation, right? So pyroptosis is with caspase activation. So when examiner is talking about pyroptosis, so pyroptosis will be with caspase activation, right? But apoptosis was also with caspase activation. So what is the difference between pyroptosis and necroptosis and apoptosis? How apoptosis differs? Necroptosis is without caspase. Pyroptosis is programmed cell death with caspase activation. But here caspases are different. That is the important thing. What are those caspases? Caspase 1 and caspase 11. Right? So that is very important to understand, and that is how future question will be framed in exam. Pyroptosis involves caspase 1 and 11, and if you remember, just now we have discussed, so I am writing here, in apoptosis which caspases were involved. If you remember, C3, 6, 7, 8, 9, 10. So these were apoptosis caspases. So always keep that in your mind. In apoptosis, caspases were different. In pyroptosis, it is C1 and C11. So this is how first we have to understand that programmed cell death without caspase is necroptosis. Programmed cell death with caspase activation is pyroptosis if caspase is 1 and 11. That is important. Right? Now we will discuss about necroptosis number 1. So number 1 topic we are going to discuss will be necroptosis. Necroptosis. If you see the name itself is informative, means it reveals information that necroptosis consists of necrosis plus apoptosis. So this is a programmed cell death which is a hybrid terminology of necrosis and apoptosis, right? So it's a hybrid terminology of necrosis and apoptosis. So what happens in necroptosis? Why it is called as necrosis plus apoptosis? What is like apoptosis? So like apoptosis, as I have told you, they are having programmed cell death, right? Now we will see what is like a necrosis. So why they are considered as necrosis? Now we will see that thing also. So to understand the necroptosis, first we should learn the mechanism of necroptosis. Mechanism of necroptosis is very interesting. Why? Because examiner can ask question कि necroptosis resembles like which pathway of apoptosis? Intrinsic pathway or extrinsic pathway? And you can answer this question only when you know the mechanism of necroptosis. Mechanism of necroptosis revolves around the death receptors. So what happens in necroptosis? Here I will tell you. tumor necrosis factor when they combine with, so this receptor when they combine with tumor necrosis factor, this will, so when tumor necrosis factor combines with tumor necrosis factor, this will activate, or you can say this will recruit, or you can say this will recruit two important proteins which are called as RIP1 and RIP3. RIP1 plus RIP3. So these are the two proteins which will be recruited by tumor necrosis factor. So when tumor necrosis factor combines with their receptors RIP1 and RIP3, these are two important proteins which will appear in the cytoplasm. What is the name of RIP1? RIP1, I just write this, receptor interacting protein. RIP1 stands for receptor interacting protein. Receptor interacting protein. So this is the full form of RIP, right? Receptor interacting protein 1 and 3. Receptor interacting protein 1 and 3 will be recruited. So what I want to tell you this, up to this is happening common to both necroptosis and apoptosis, right? So up to this stage, this will be happening for both apoptosis and necrosis. See this? Both apoptosis and necrosis. Here onwards, their pathway will diversify. So what I want to tell you, after this RIP1, RIP3 inclusion, there are two possibilities. Right side? What I am telling, there are two important possibilities. What are these possibilities? RIP1, RIP3, either they will be accompanied by caspase 8 or they will be having no help from or no involvement from the caspase 8, right? So when they had involvement from the caspase 8, see this, what I am writing here, when caspase 8 is involved here along with caspase 8, then this will be undergoing to apoptosis. You remember, extrinsic pathway of apoptosis. So with caspase, apoptosis. That is why I said without caspase 8. So when there is no caspase 8, then what will happen? Without caspase 8, this will put oxidative stress on the mitochondria. So mitochondrial stress. So mitochondria will be under stress. Because of the mitochondrial stress, as we have learned earlier, free radicals will be generated, right? Free radicals will be generated. And what are the targets of the free radical? What are the targets of the free radical? They will damage the cell membrane, right? So because of that, you will see, number one, there will be cell swelling, right? So there will be cell swelling. Free radicals will be causing cell membrane damage. Cell membrane damage. So cellular swelling, cell membrane damage. And third, you will see, because membrane has been damaged, so there will be presence of inflammation. Presence of inflammation. So cellular swelling, cell membrane damage, presence of inflammation. These are the morphology of necroptosis, right? So that is how necroptosis differs from the extrinsic pathway. Extrinsic pathway of apoptosis, when caspase 8 was involved. But in case of necroptosis, when there was no caspase 8, it has put stress on the mitochondria, and as a result, free radicals were produced. And free radicals which has been produced, they had created all these morphological changes. Cellular swelling, membrane damage, inflammation. Now my question is, where you will find inflammation? Where you will find cell membrane damage? Where you will see cellular swelling? In apoptosis or necrosis? Necrosis. That is why, because of this morphological spectrum, now you can see, when we were discussing about, I said necroptosis is a hybrid terminology of necrosis. Hybrid terminology of necrosis and apoptosis. Apoptosis, programmed cell death, right? But why it is called as necrosis? Because of the morphology. What is the reason? Morphology of the cell will be like this. What is the morphology? Morphology of cell is showing cellular swelling, membrane damage, and presence of inflammation. That is why when examiner will ask you this question, ki what is necroptosis? So these are changes of necrosis. So that is why they are considered as variants of necrosis. So that is very, very important point which examiner can ask in future exam. variants of necrosis. Necroptosis are variants of necrosis. Why variants of necrosis? Because of this morphological presentation. Why it is variant? Why not it is necrosis? This thought gives us another insight about this process or about future MCQ. Necroptosis is variant of necrosis. Why? Because necrosis is always pathological. You remember when we have discussed, I told you necrosis is always pathological, but necroptosis. So that is the future question. So here we must understand that necroptosis is both pathological and physiological. So necroptosis, this is how it differs from the necrosis. It is both physiologic and pathologic. So it is both physiologic and pathologic. So necroptosis is both physiologic and pathologic. What is physiologic example? What is physiologic example? Mammalian bone growth plate formation. So here example is mammalian growth plate formation. So mammalian growth plate formation in the bone, right? So mammalian bone growth plate formation. So this is the physiologic example. So that is the important point. Physiological example of necroptosis, mammalian growth plate formation, and pathological there are several examples. For example, just now we have discussed neurodegenerative disorders, right? So whatever neurodegenerative disorders, we know that Alzheimer's disease, Parkinson's, Huntington's, these are all pathologic example. And second example is the pancreatitis, right? So these are examples of the necroptosis. So now we can understand necroptosis is a variant of necrosis precisely. Why not necrosis? Because necrosis is always pathological, but necroptosis can be physiological also, and this is the example of the physiological. So keep that point in your mind, can be a future exam question, right? So this can be also a future exam question. So that is why we have to keep all these unique points in our mind for future exam point of view, right? So we have discussed about one, programmed cell death without caspase is necroptosis. Now second we are going to discuss is the pyroptosis. So second thing we are going to discuss pyroptosis. If you look at the division of this name, pyroptosis, right? So this name says that it is pyrogen induced apoptosis, which is a quite contradictory statement, right? Because pyrogen is an inflammatory cytokine, right? What is pyrogen? Best pyrogen is IL-1, right? So pyrogen is an inflammatory cytokine, and if there is an inflammation, then how come it can be apoptosis? Simple, right? So but you have to understand this name had been coined because initially they thought it is a pyroptosis, or it should be considered as. Why it is pyroptosis? Why it is considered as apoptosis like? Because as again I will tell you, because they are having programmed cell death, right? So pyroptosis is also a variant of necrosis. So here also we have to remember pyroptosis is nowadays considered as variant of necrosis, right? Mechanism of pyroptosis is little complicated and very, very important because it's not given very clearly in many of the textbooks also. So here I want to tell you about the mechanism of pyroptosis in detail. Mechanism of pyroptosis, it can be any bacterial antigen which will induce pyroptosis. Bacterial antigen, basically here I will give you example of the one bacterial antigen which is called as flagellin, present in the flagellated organism like salmonella, right? So I am taking example of the flagellin. So just keep this in your mind, flagellin. So whenever bacterial antigen will come and attack our cell, then what will be the sequences of the pyroptosis I am going to demonstrate, right? Suppose this bacterial antigen has arrived. This is the bacterial antigen, and this one is the cell architecture, right? This is the cell which I am going to demonstrate here, and this is the plasma membrane, right? This is the plasma membrane, and just two things, just for the illustrative purpose I am making it as a lipid bilayer, right? So plasma membrane, so this bacterial antigen or flagellin they will act at two places, at one plasma membrane and second inside the cytoplasm for induction of the pyroptosis with the help of caspase 1 and caspase 11. You remember programmed cell death with caspase 1 and 11. So what happens whenever this bacterial antigen will come? So first they will meet a membrane receptor which is called as toll like receptors, right? So these are toll like receptors. So toll like receptors are present on the membrane. So whenever this bacterial antigen will combine, see this, whenever this bacterial antigen will combine with toll like receptor, what will happen? This will send signal to the cytoplasm, and in the cytoplasm this will activate procaspase 11 to caspase 11, right? So procaspase 11 will be converted into caspase 11 by the toll like receptor, right? So this activated caspase 11 will be having route of action. They will be causing cell swelling, right? So they will be causing cell swelling, cell membrane damage, and because of the cell membrane damage there will be presence of inflammation. So now you can see that morphology is similar to the necrosis or necroptosis, right? Caspase 11 was activated with the toll like receptor, and because of this caspase 11 activation, mitochondrial stress will develop, free radicals will be produced, there will be cellular swelling, cell membrane damage, and presence of inflammation, just like the necrosis or necroptosis, right? This is the one event. Second pattern of event you can observe. See here, bacterial antigen. See this is the bacterial antigen, right? This is the bacterial antigen which I have demonstrated on the membrane surface, but this bacterial antigen can come directly into the cytoplasm also. So again I am showing you, this is the bacterial antigen or flagellin. Flagellin has entered into the cytoplasm. Into the cytoplasm, what will happen? Into the cytoplasm, there is another receptor which is called as nod-like receptor, NLR. This is called as nod-like receptor, right? So this is nod-like receptor which is present inside the cytoplasm. This nod-like receptor antigen combination within the cytoplasm will activate. So now this combination will activate inflammasome. This is the another important feature. When nod-like receptor will combine with antigen, this will activate inflammasome. And this inflammasome activation will activate procaspase 1 into active form of caspase 1. So this is how you will see the changes in a cell, right? So when antigen will come into the cytoplasm, they will combine with nod-like receptor which will activate inflammasome, and inflammasome will activate procaspase 1 into caspase 1. This caspase 1 is very important, and this was the question also in this all India exam. Inflammasome, when they will activate caspase 1, what is the function? So this caspase 1 will activate interleukin 1, right? So this will activate interleukin 1 and interleukin 18. So these two interleukins are called as inflammatory cytokine, right? And they will also cause the same thing: cell swelling, cell membrane damage, and inflammation, right? So these are the same thing. So now we can see the scenario. Antigen comes into the cytoplasm, combines with nod-like receptor, activates inflammasome. Then procaspase 1 is getting converted into caspase 1, and IL-1 and IL-18 is formed, which will cause cell swelling, cell membrane damage, and inflammation. So same like necrosis. That is why I said in the beginning that pyroptosis is a variant of necrosis nowadays because of these features, right? Here I want to tell you one more important point. and that is this caspase 11. This caspase 11 has the tendency to activate procaspase 1 activation to the caspase 1. So this caspase 11 can activate procaspase 1 into caspase 1. So this is another important interlink between caspase 1 and 11, right? So caspase 1 was the question in this All India exam. IL-1 is activated, or caspase 1 will activate which of the following cytokine. So caspase 1 will activate which of the following cytokine? Answer was interleukin 1. So answer was interleukin 1. So future exam, interleukin 1, interleukin 18, both of the above. Answer should be both of the above. That is how we have to predict the future question, right? So this is about pyroptosis, and pyroptosis is also a variant of necrosis, right? So these are two programmed cell death which is important for conceptual understanding also and for exam also. If you remember, when we were discussing about cellular adaptation, we said it's a altered physiologic stimuli or non-lethal stimuli, then lethal stimuli causing cell injury, and then we discussed about the chronic injurious stimuli leading to the intracellular accumulations, right? So now we are going to see that intracellular accumulations because of the chronic lethal stimuli or chronic injurious stimuli. So when we talk about chronic injurious stimuli, what happens? Chronic injurious stimuli leads to intracellular accumulation. As I said, intracellular accumulations. So what are these intracellular accumulations? So now question comes to our mind, in chronic injuries, that intracellular accumulations, what are these intracellular accumulations? What are these intracellular accumulation? It can be normal substances also, which we see in our human body normally, right? Or it can be abnormal substances also, and it can be calcium deposit also, which is called as calcification. So after the cellular damage there will be calcium deposition, that is called calcification, right? So what are the normal substances we can see, right? What are the normal substances we can see? So now I will tell you about this normal substances. Normal substances, as I have told you, normally they will be present, but during chronic stimuli they will be having excessive accumulations. It can be certain substances, it can be certain pigments also, right? So it can be substances as well as pigments also. So what are the substances we will see? We will see the substances like lipid, glycogen, proteins, right? So lipid, glycogen and protein. So these are the some of the normal substances which will be accumulated, right? What are the other important substances which can be normally present in our human body? It can be pigments, right? Which pigments we have to understand? So in pigments, these are lipofuscin, or also known as lipochrome. It can be hemosiderin, or it can be melanin. So these are some of the example. I'm just mentioning few of them. So because of the chronic stimulus or chronic injurious stimuli, lipid, glycogen, protein or pigments, lipofuscin, hemosiderin can be accumulated within our human cells, right? Abnormal is the from outside environment, like coal, right, like silica. So all these are abnormal, and calcification we will discuss in the end, right? So this is how we are going to discuss about the normal and abnormal accumulation within the cytoplasm. So first I will talk about the normal substances like lipid, glycogen and protein. So question number 1, what examiner may ask you in exam, which is the most common intracellular accumulation in the human body? Most common intracellular accumulation in the human body. So remember, lipid is the most common intracellular accumulation in the human body. This is the older question also in state exams, right? So intracellular accumulation, lipid, right? So first we will start from the lipid. Lipid is the most common intracellular accumulation, as I have told you. This is the most common intracellular deposits, right? Most common intracellular deposits within the cell. This intracellular accumulation of lipid is also known as steatosis. That terminology you will often see in your liver chapter, which we will discuss, right? So steatosis is the excessive fat accumulation or excessive increased lipid deposits. Increased lipid deposits within cells are called as steatosis, right? So steatosis, increased lipid deposit. Which lipid will be deposited? Because lipid will be deposited in various forms. It will be triglyceride, right? It can be triglyceride, or it can be cholesterol, or it can be cholesterol esters, right? It can be phospholipid. So these are the various forms of the lipid which will be accumulated within the cell. Now the question examiner is asking, कि in steatosis or in fatty change, which is the most common accumulation? Which is the most common accumulation? Triglyceride, cholesterol, or esters, or phospholipid? All India 2014, All India 2016. So always remember, triglyceride is the most common. Triglyceride is the most common lipid accumulation. So that was the answer. Most common lipid accumulation is the triglycerides. So most common lipidic accumulation in steatosis is the triglyceride, and I will tell you how examiner will be asking, or how examiner had asked this question in All India exam. In All India exam, this had been asked in context with non-alcoholic fatty liver disease. Non-alcoholic fatty liver disease in fatty change of the liver, which is the most common accumulation. So examiner can ask this question like this also: in hepatitis, whenever there is a fatty change, which fat accumulation is the most common? Triglyceride. So NAFLD, hepatitis, whether it is alcoholic or viral whatsoever, in all format of hepatitis or fatty change, triglyceride is the most common substance which will be accumulated, right? So that point we have to keep in mind. And how we are going to confirm that this is the lipid? See how it will be looking like, and I will tell you. When you are going to see a cell, there will be a nucleus, right? This is the nucleus, and cytoplasm will be having vacuoles, clear vacuoles. So these clear vacuoles are lipid accumulation, right? So these clear vacuoles can be lipid also. This can be another substance like glycogen also. How we will know that this is lipid only? How we will know that this is lipid only? So to know that this is a lipid only, we have to use special stains for the lipid. So what are the special stains for lipid? The special stains for lipid will be, number one is oil red O stain. So oil red O stain is the special stain which we use for lipid. Another important stain is the Sudan black B. So Sudan black B, or simply Sudan stain, is used for the fatty tissue, right? So here we have to understand what are the special stains for the lipid: oil red O stain and Sudan black B. Easy to remember. What will be color of the lipid? When we are seeing oil red O, see the name itself is dictating what should be the color of lipid? Red color. Lipid will be red or orange, whereas Sudan black B, color of fat will be black. So fatty tissue will be black, whereas in case of oil red O stain, it will be red or orange color. This is how we will identify. I'll just show you two of the images of the lipid accumulation. See this is the liver biopsy, and in this liver biopsy, you can notice there are big vacuoles, right? These bigger vacuoles are having fat, right? These bigger vacuoles are having fat, or you can say lipid. So how to identify this fat? You can see this fat, this one, this cell. In this cell you can see there is a fat, and there is a nucleus which has been pushed to the periphery because of the lipid, right? So how we will know that this is the lipid? So to identify this lipid, we have to apply special stain. See, when we apply special stain, right, then we see red or orange color of accumulation. So this is oil red O stain. So that is why I said the name itself is the informative name, oil red O stain. Oil red O stain, lipid will be red or orange in color. As you can see, these are the lipid which will be red or orange in color. Oil red O stain, this was the question in AIMS 2016. This is the best stain to identify lipid on frozen section, right? So remember, this is the best stain. Oil red O stain is the best stain to detect lipid, where? On frozen section. Frozen section is also called as cryo section, right? So frozen section or cryo section, frozen section is also known as cryo section. So examiner had used the name cryo section only. So examiner question was on cryo section, to determine the lipid, what will be the best stain? So answer was oil red O stain. So oil red O stain is the best lipid stain for detection of lipid on frozen section, and they will be looking red color, and that point we have to keep in mind. And suppose when you are getting a biopsy like this, liver biopsy, and you are noticing numerous black color deposit, so always remember this is the lipid on Sudan black B staining. So this is the Sudan black B staining. Sudan black B stain. Sudan black B stain, we will see black color of the pigment, of the lipid pigment, right? This is how we will see two important thing, right? Now you can see oil red O red color, Sudan black B, it will be black color, right? So this is about the lipid accumulation. Second important accumulation is the glycogen. So whenever there is a chronic injurious stimuli, some of the cells will be having glycogen accumulation, right? Glycogen accumulation will be also presenting in the same way. You will be seeing a cell, there will be a nucleus, and you will be noticing vacuoles. So these vacuoles are having glycogen. How we are going to confirm? By using a special stain. So what is a special stain for glycogen? A special stain for glycogen is PAS stain, pyridic acid SCIF. So they will be PAS positive, number one, and second, best carmine stain. So these are the two important stains, best carmine stain. So PAS stain or best carmine stain, on both you will see glycogen will be looking red in color. So this glycogen will be looking red in color on examination, right? So whenever you will see glycogen on PAS stain or best carmine stain, they will be appearing red granules. So this is how we will see glycogen accumulation, right? Third one, third important accumulation is about protein. That is important. Proteins accumulation, why I wanted to discuss this? Because recently in All India and AIMS both exam, many questions had been asked from this area, right? So whenever there is a protein accumulation, how we are going to understand about this protein accumulation? What is the reason of the protein accumulation, right? So whenever there is a excessive protein formation or excessive protein synthesis, this excessive protein synthesis will be undergoing increased misfolding of the protein. Misfolding of the protein. So because of excessive protein, there will be misfolding of the protein, and when misfolding of the protein is going on, then in normal response. So see, what will be the normal response? First I will tell you what will be the normal response to the misfolding of the protein or to the excessive misfolding of the protein in our human cells or a human body, right? So normal response will be unfolding protein response. This is called as UPR, right? This is called as unfolding protein response, right? This is called as unfolding protein response. So they will be trying to unfold this protein. How they will be trying to unfolding protein response? What are those mechanisms? So that is important. That is important to concentrate. What are the unfolding protein response? In this unfolding protein response, number one, Because of the misfolding of the protein, there will be stress on the endoplasmic reticulum, right? So this will cause stress on endoplasmic reticulum. And whenever endoplasmic reticulum is stressed because of excessive misfolding of the protein, they will start synthesizing chaperones. So that is very important to understand. Because of this stress in endoplasmic reticulum, they will synthesize chaperones, right? Chaperones are the proteins which are required for proper folding. That was the All India question in 2017 and 18. Proper folding of the protein. So they will be causing proper folding of proteins, right? So that is the function of chaperone. So that was the question in exam. Okay, what is the function of chaperone? Function of chaperone is to induce proper folding. So whatever was misfolded, which has put stress on the endoplasmic reticulum has synthesized chaperones which are involved in proper protein folding, right? This is one important mechanism, right? Second possibility, they will be inducing another programmed cell death, which is called as autophagy. There is another important programmed cell death which I wanted to tell you here, is called as autophagy. So autophagy is also a example of programmed cell death. So autophagy is also an example of programmed cell death, and what they do, whenever there is a excessive protein, intracellular protein degradation system will be activated. You remember during atrophy also we have discussed about this, so I am not going to discuss very much detail about this thing. So whenever there is a excessive protein, what I said, this will be excessive protein misfolding, I should say. This will activate intracellular protein degradation system. Intracellular protein degradation system. So what happens in this intracellular protein degradation system, right? So this will induce ubiquitin. Ubiquitin will combine with the protein, right? So whatever target protein, this was the misfolded target protein, I should say, target protein was there. So this target protein will be then it will be taken in a double membrane size called autophagosome. So this is called autophagosome, and this autophagosome will combine with lysosome, right? So this will combine with lysosome, and lysosomal enzymes will cause degradation of these proteins. So this is the autophagy, right? This is the autophagy concept we have discussed in atrophy also. So whenever there is a excessive misfolding of the protein, ubiquitin will be synthesized, which will combine with target protein. They will form autophagosome, they will combine with lysosome, and by this process target protein will be removed, and this is called as self-eating or autophagy, right? Third thing, whenever there is a excessive protein synthesis, third thing will be this excessive protein synthesis will induce caspase, right? And this caspase will remove the protein by apoptosis. So apoptosis will remove the excessive misfolded protein. So this is how unfolding protein response works. So whenever there is an excessive protein, this will be removed either by the chaperones by doing proper folding, not removed, they will be properly folded, or they will be removed by autophagy, or you can also say that it can be also removed by ubiquitin proteasome degradation pathway, which we have read in atrophy, right? And third, they will induce caspase to induce apoptosis of the misfolded protein. So this is how it works in our human body. And when this will fail, so when this unfolding protein response, which we started, you remember, unfolding protein response. So when this unfolding protein response will fail, then you will see misfolding protein disorder. As I have discussed, Alzheimer's disease, right, and Parkinson's whatever neurodegenerative disorder we have seen, right? So this point we have to remember for the protein, right? Here I want to emphasize one important thing, and that emphasis is for autophagy. So what is the implications of the autophagy? Means autophagy or ubiquitin proteasome degradation pathway, right? So I wanted to tell you one important point which was questioned in All India exam as well as DNB exam this year. Autophagy ubiquitin proteasome degradation pathway is associated with, question is associated with. So to whom they are associated with? Whom they are associated with? They are associated with these following things. Remember, atrophy, right? If you remember, we have discussed all these intracellular degradation pathway in atrophy. They are associated with atrophy. They are associated with inflammation. Inflammation. Which inflammation? Can be a future question. Which inflammation? A very important inflammation, especially for the Indian territory, right? That is the TB. Inflammation, especially the TB, because this is the one update in Robbins which examiner may ask in your exam. In TB, what is the autophagy association? There is a gene called ATGS5 gene. ATGS5 gene controls the normal phagocytic function of macrophage, right? So ATGS5 gene is a gene present in our human body which controls the phagocytic function of the macrophage via autophagy, right? But when there is a deletion, of the ATGS5 gene deletion, right? So this macrophage function will be defective, right? So because of deletion, this will become defective, and that is why this patient will be having increased susceptibility for the TB inflammation. So that can be a future question. Increased susceptibility of the TB inflammation is associated with which of the following genes? So answer should be ATGS5 gene. So many of the research article has been published in support of this one. ATGS5 controls the macrophage function, and whenever there is a deletion in ATGS5, susceptibility to have more tuberculous inflammation will be there, and that is the important thing which we have to keep in mind for future exam. So atrophy, inflammation. Third is atrophy, inflammation, and also associated with cancers. And in cancers, autophagy is dysregulated. This point we have to keep in mind, because in cancers, autophagy means what? Self-eating. So self-eating can remove the cancer cell also, but what is happening? Autophagy is dysregulated, not the normal one. So they will become abnormal. So means there will be reduced autophagy in cancer cells, right? And the fourth one, which was the DNB question, autophagy or ubiquitin proteasome pathway is associated with mitochondrial inheritance. In genetics, we will see that, right? In genetics, I will demonstrate that how it is associated with mitochondrial inheritance. So these are the four important points we have to keep in our mind. The autophagy is associated with atrophy, inflammation, cancer, and mitochondrial inheritance, right? This is very important for All India exam. So we have to keep that in our mind. So this is about intracellular protein accumulation. Now we will see about the pigment accumulation. So next topic we are going to see is the pigment, right? Pigment accumulation, right? Pigment accumulation, I will say normal pigments, and second will be abnormal pigment. Normal and abnormal. Normal pigment, we all know that, as I have told you just now, lipofuscin, hemosiderin, and melanin, right? Lipofuscin, hemosiderin, melanin, they will be present in normal also, and they can be present in abnormal also, right? So when they are under normal, no problem. They may be a sign of some. So now we will discuss about next topic. Next topic we are going to discuss about the pigment. right? So what are the pigments accumulation we are going to discuss here? Here I would like to demonstrate like this thing. Okay, there are two colors of the pigments. To make it more simpler, I want to say that there are two colors of the pigment. One is the brown pigment in our human body, brown pigment in our human body, brown pigment, and second I will tell you black pigment. So there are two types of pigment we often notice, right? Brown and black pigment. What is the differential for the brown pigment? So here, differential for brown pigment, there are two pigments which we'll discuss here. One is the lipofuscin, and second brown pigment is the hemosiderin. So lipofuscin and hemosiderin. So these are the two pigments, lipofuscin and hemosiderin. These are the two brown pigment. Black pigment in human body is the melanin, right? So this black pigment in human body is the melanin. Melanin is present in our normal skin also. It is present in our normal skin also, and this will be present in melanoma also. Right? So that is the black pigment, melanin. Question in All India exam was, melanin, which special stain we use? What is the name of special stain for melanin? That was the question in All India exam, right? So melanin is detected by which special stain? Answer is Fontana-Masson stain. Fontana-Masson. So we must remember this name. Fontana-Masson is the special stain for melanin. That was the right answer in All India exam, right? So this is the right answer in All India exam. Fontana-Masson is used for melanin, right? So this is about black pigment. Now we will focus on the brown pigment, right? So one by one we will discuss. First, we will see what is lipofuscin. First, we will see what is lipofuscin. Lipofuscin is also known as aging pigment. So that is the important thing we have to remember. Lipofuscin is also known as aging pigment because they are normal pigment which is formed in our human body because of the free radical damage. Because as you remember, when we were discussing about the free radicals, I told you normally free radicals are produced in our human body because of the oxidative stress on the mitochondria. So that free radical, see this, what I am showing you, that free radical which is produced in human body will act on the cell membrane, right? This is the cell membrane, this is the nucleus, right? Will act on the cell membrane and they will damage the phospholipid part of the cell membrane, and this brown phospholipid part will be accumulated around the nucleus, right? This will be accumulated around the nucleus. So this is the aging pigment, and this aging pigment, what is the color? This is the question which examiner asked in All India exam and DNB exam. Brown pigment. Brown pigment, what is the location? Around the nucleus, perinuclear. So these are the perinuclear brown pigment. So what is lipofuscin? Aging pigment, brown pigment, perinuclear location, right? Because of the free radical causing damage of the cell membrane. So if cell membrane has been damaged, so automatically we can understand what will be the constituent of the lipofuscin. So lipofuscin is made up of, if it is a cell membrane, so it will be made up of phospholipid, right? So this is the phospholipid plus proteins. So phospholipid is the dominant thing which will be present in the lipofuscin. So brown pigment, perinuclear pigment consisting of the phospholipid caused or formed by the free radical damage. This is called aging pigment, right? But here what we need to notice that it is not only aging, right? It is not only aging where you will find this pigment. This pigment is also seen in certain pathological condition like cancer cachexia. Cancer cachexia can be also seen in severe malnutrition. Severe malnutrition. So this was the All India question in 2016. Question was, lipofuscin is present in which of the following condition? Options were aging, cancer cachexia, and severe malnutrition, all of the above. And now you can see that for all of them lipofuscin will be present. So lipofuscin is an aging pigment which will be present in aging also and cancer cachexia, severe malnutrition also. and they are formed by free radical damage. That is why there is a nickname which we use for lipofuscin. These are the telltale sign of free radical damage. So there is a phrase used for lipofuscin, telltale sign of free radical injury, right? So whenever you, what is the meaning, Hana? What is the meaning of this phrase? Telltale sign means lipofuscin is telling that my membrane, cell membrane was damaged, and from the membrane phospholipid this brown perinuclear pigment has been formed. So telltale sign of free radical injury is lipofuscin, or also known as wear and tear pigment. Wear and tear pigment. So lipofuscin is a brown pigment. You remember we discussed like this. Lipofuscin is a brown pigment. Brown. So the question is how to differentiate this brown pigment from lipofuscin, the brown pigment, from another brown pigment in our human body, that is called hemosiderin. Right? So how to differentiate between the lipofuscin versus hemosiderin? So to differentiate between lipofuscin and hemosiderin, we require a special stain. Which special stain you are going to use for lipofuscin? See, the name itself is informative. Lipid, it is made up of lipid. So we are going to use lipid stain, oil red O stain or Sudan black B stain. Sudan black B stain, right? And already we have discussed it is red in color, oil red, Sudan black will be black in color. Now the question is, this hemosiderin brown pigment, how we are going to discriminate from the lipofuscin? Hemosiderin is having iron. Iron, storage form of iron is Fe3+. Always remember, storage form of iron is Fe3+ micelles present in the storage form of iron, right? So what stain we are going to use for hemosiderin is Pearl's stain. Pearl was name of the scientist, and this was the question in one of the older All India exam. Pearl's stain is used for hemosiderin. That was the simple question. Pearl's stain is used for hemosiderin. Pearl was the scientist, as I said, who has discovered the name of a stain, which was stain for the iron, which is known as Prussian blue. So it is also called as Pearl's Prussian blue stain. Pearl's Prussian blue stain, color of iron will be blue. So Pearl's Prussian blue stain will detect iron in Fe3+ form, and they will be appearing blue in color. So that was the question in AIMS 2016, where they had shown one image of the liver biopsy. It was hemochromatosis, and the pigments were looking blue color, and they had asked what is the name of this special stain. So that is Pearl's Prussian blue stain. Hemosiderin will be looking blue in color, right? For example, look at these diagrams which I wanted to show you. Look at this picture. This diagram, if you see, this is the liver biopsy. In this liver biopsy, you can see there is a cell, and you can see this cell which I wanted to show you. In this cell, around the nucleus, this is the nucleus. Around the nucleus there are brown pigment. There are brown pigment. So these are perinuclear brown pigment. So what are these brown pigment? Question is, what are these brown pigment? So always remember, when examiner asks question like this, perinuclear brown pigment in the liver biopsy, and no history is given, your mind should work like this. It can be either hemosiderin or lipofuscin, like this. Either hemosiderin or lipofuscin. So we have to decide whether it is lipofuscin or hemosiderin. So if examiner is saying to choose one better answer, so always remember lipofuscin is the most common accumulation, because in aging also it will be seen. But when examiner is mentioning those special stain, oil red O positive, Sudan black B positive, think about lipofuscin. When examiner is saying it is Pearl's Prussian blue stain, then you think about hemosiderin. So see, this is how examiner can ask question. Okay, there was a perinuclear brown pigment, and this is the staining. So it is lipid, right? Red color, oil red O stain. When they are showing this black color, Sudan black B, it is again lipid. And when examiner is showing like this, where you are noticing these pigments are blue color, so this is the Pearl's Prussian blue stain, which was the question in AIMS exam also. So Pearl's Prussian blue stain, you can see iron will be blue. So this is the Pearl's Prussian blue stain. So Pearl's Prussian blue stain, iron will be blue. You can see the name itself is informative. Pearl's Prussian blue, iron will be blue. Right? So this is how we can understand the pigments. Now we will see the last thing in the pigment accumulation was the calcification.